All right, this is the second part of the prokaryotics PowerPoint section that I'll review with you guys. So we finished up with the cell membrane, and so now we're ready to move to the inside of the prokaryotic cell or the bacterial cell. So the first internal structure is going to be the cytoplasm. And you can see dense gelatinous. Gelatinous, think of Jello or think of runny Jello, right? It's that's probably a good idea to think of it about it, and maybe not even runny Jello, somewhere between solidified Jello and runny Jello. That's kind of where it's at, right? A large amount of water there, and our cells have that as well. Internal structure. The next thing is going to be the chromosome, and this is going to be the DNA of the bacterial cell. Remember, all cells have DNA. Oh, I'll close that. Close my email so it doesn't pop up. Okay. All cells have DNA. Bacterial cells are cells, right? The DNA of a bacterial cell, they only have one chromosome, and it's in a circle, and it is double-stranded DNA, right? The eukaryotic chromosome, like I say, our chromosomes, for example, are kind of paired together, so they typically looks like an X, and we have 23 pairs or 46 chromosomes, right? So compare that versus bacteria, one, and it's in a circle, right? And that has everything the bacterial cell needs to live its life and reproduce and make another cell, right? And it's coiled together. And you'll see this in a figure, right? And so what this figure is showing you, this is in yellow, right? That's all the DNA that's been coiled together. So it's not stretched out into a big circle, right? So the way you can think about that is that it's like a rubber band. It's a big rubber band's a big circle, but then it coils up on itself. So that's what you're seeing here. Right? Other internal structures, a plasmid. Plasmids are also circular DNA, but they're much smaller than a chromosome. And you can see in red there, they're not essential, meaning they're optional. Some bacteria have them, some bacteria don't have them, okay? They can be replicated, they can be shared, but they're optional. If a bacterial cell does have plasmids, they usually give them some advantage, right? Whether it's resisting a drug, resisting some kind of something that's toxic, making some special enzyme or some special toxin. Right, so it's usually a good thing for the bacteria, maybe a bad thing for us, these plasmids, okay? But they're optional. Next internal structure is going to be the ribosome. Right? And all cells will have ribosomes, including bacterial cells. In that third little dash there, the size difference, okay? That size is going to be important as well. I'll show you that in the figure in a moment. Ribosomes are the site of protein synthesis. Right, that's what's missing. And then we'll come back to this in future chapters. Right, this idea of protein synthesis taking part the ribosome. You can almost think of it as like a little factory or a little assembly line. Is this ribosome? This is a figure showing you the ribosome and showing you the size of it. So it's 70S. The prokaryotic or bacterial ribosome is 70S. But if you look at the figure, it says 30 plus 50 to give you 70. Well, that's because the measurement S is not actually a measurement of size. It's kind of weird. It's actually a measurement of speed or time, but it's called a Svedberg unit. But we won't get into that too detailed, and I won't ask you about the large and small subunits, but I do want you to know the size of the bacterial ribosome is 70S. And that's because the eukaryotic ribosome is going to have a different size, and that difference does matter actually when it comes to treating infections and how some of our antimicrobial drugs work. So that size difference is important. And your book does talk about other internal structures, but I'm not going to test you over them. So the next thing here is going to be endospores, and they're kind of internal structures, they're kind of not, but we'll cover it here. So endo does mean in, so it is inside the cell, but they're going to be special. Okay, Endospores are, you can see, inert means they have no metabolism and they're not reproducing. Resting cells produced primarily by Clostridium and Bacillus. There's other groups, but I just want you to know these main two here. Okay. And there's a life cycle associated with these spores that we'll see in the figure, which is sporulation, making a spore, and germination, coming back to a vegetative state. And so what happens, you have a cell, and then all of a sudden, let's say, not all of a sudden, but let's say you run out of water or the bacterial cell runs out of nutrients. It says, oh no, instead of just dying, I need to preserve my DNA. And so that's what it's about. It's about survival and preservation of the DNA. So replicate the DNA, divide off, all right, form a little spore, all right, form a little what's called a cortex here. There's your endospore. And then it's ready, it's good to go, but it doesn't stay like this because kind of what's called the mother cell here dies off, and you're left with this free spore. And that can just hang around for weeks, years, decades, right? Centuries, and just hanging out with the DNA inside, right? Because remember, all it is just chemicals. There's no metabolism, nothing's being used, nothing is being broken down. But then you have water, you have nutrients, germination can take place, and then boom, the bacterial cell can start growing again. Right, here you go. Dehydrated, metabolically inactive. Scientists believe they found endospores that are millions of years old. I think they were like in glaciers or something like that, very cold temperatures. Very tough to kill, right? So boiling doesn't work. Your Lysol, bleach doesn't work. You usually have to use higher end chemicals or like pressurized steam, like a pressure cooker to kill them. And so they're really good at survival. Right? So that's the key thing there. And then the last thing that we'll get into then is shapes and arrangements. Right? So bacteria generally have three basic shapes. Again, there's some exceptions to this that we won't necessarily get into, but there's three basic shapes. The cocci, which is shaped like a sphere. The bacillus, which is a rod. And under the bacillus, you can see there's some kind of special names you should be familiar with, which is the coccobacillus, which looks like a bean. Very short and plump, but still a bacillus, so it's a coccobacilli. The vibrio, which is kind of like a cashew. It's a bacillus that's curved. And then you have your spirillum / spirochete shape, which is like a twisted rod or a spring. Then with arrangement, okay, this is how they can be put together. Right? Notice only cocci and bacilli have arrangement. There's no arrangement for spirochete, so it means they're just going to be by themselves. So your cocci, you can have them alone, or you can have diplococci. That shows you a picture. You can have a tetrad. All right, you should be familiar with the tetrad. Tetra means four, or tet means four. So it's a group of four together. So if you put two diplococci, like one on top of another, that would be your tetrad. Right? Irregular clusters. Clusters is staphylo. So staphylococci. Right? Chains. The prefix for chains is strepto. So streptococci. You can see at the bottom. Right? The cubical packets, don't worry about that. Bacilli, diplobacilli, it's pretty easy. You can have chains, so that would be streptobacilli, right? And then palisades is kind of like a hinge-shaped bacteria. I'm not really worried about that one, but you should be familiar with the others. And be able to recognize bacterial shapes on an exam, and be able to recognize bacterial arrangements on an exam, like the actual figures, right? And that's it for this chapter. So there's really only two sections, right? So this one was internal structures, endospores, and shapes and arrangements.